Okay, so I don't do very much by way of ontology mapping, and the synergy with other ontologies for Pro is most typically expressed in terms of reuse. However, we do have quite a bit of interplay with various protein databases, and this has led to us having an interest in ontology mapping, at least on a small scale. So it's more as a consumer of such mappings that I give this talk. So I'd like to start by providing a bit of background on Pro. In the first slide, touching upon a bit of the reasoning behind why it was created. Here you see a number of different protein entities related to SMAD2, pictorially represented in the first column. The second column gives a quick description, while the third indicates a few select Go terms that can apply to each. You'll note that the annotations in that Go column aren't uniform among these entities, implying that it would be useful to have targets of annotation more specific than what was classically done years ago in the gene ontology, which is at the level of the gene. So all of the information was attached to the gene, and here provides a rationale for splitting that apart into something more specific. As indicated in the last column, UniProtKB provided a bit more specificity when it comes to sequence level differences. You see different identifiers there, -1, -2, and then a variant indicated there. And indeed, the relevant entry refers to one of these by description, the one that, so it has these three with distinct identifiers. It refers to this one in the text somewhere. And then there's additional information that you can infer these other two based on information that's in UniProt, but these aren't mentioned specifically. In other words, they don't mention TGF beta receptor plus ERK1 phosphorylated in a direct sense. So that's where Pro steps in the idea being to create accessions for protein entities at any level of specificity. So in the next slide, I want to give a broader overview of Pro while simultaneously providing a bit of history of its development. So at the very start of Pro, we intended to follow the Go paradigm and not cover species-specific proteins. So each of the classes shown here are species neutral. For the sake of understanding, we divide them into broad categories that we term levels of distinction. This, family, gene, sequence, modification. These are different levels of distinction. I'll tell you what that means in a moment. So at the gene level of distinction, any two entries at this level represent all protein products translated from a distinct gene. Now, despite this species neutral aspect that I just mentioned, it's sometimes helpful to think of the gene category in terms of a specific species. So for example, human IRF5 differs from human IRF4, IRF3, etc. But these related proteins can be subclassed under the same family level term, IRF. Now moving toward the more specific, down here in this direction, we have proteins from the same gene that differ based on the encoding mRNA, or perhaps a start site selection, or the allele, resulting in sequence differences even within a given organism. So you can see here a simple example, IRF5, isoform 1, and isoform 2. So it's two different isoforms, and those are distinct sequences. Now, finally, a given translated protein can have various co- or post-translational modifications. And this particular example here shows what is, because they're both called phosphorylated 1 here, but they're two different isoforms. So these are distinct proteoforms. We would call them distinct proteoforms, and those are under the modification level of distinction. Now, at the time that we first started Pro, we distributed a mapping file that basically indicated which UniProtKB entry would be assigned to which gene level proterm. Eventually, by user request, hi Chris Mongol, we directly included proterms based on UniProtKB entries, thereby introducing species specificity, and we did it at all levels. The orthologous genes, we have orthologous isoforms, and we have orthologous proteoforms. Another addition later still, and again by user request, was the inclusion of protein complexes. I think I forgot to mention here, the addition of the UniProt-based terms resulted in these orthologs that you see here, and they both map to the same parent species neutral term. Again, so go back to complexes. We later on added complexes. We initially left those out because Go handled them, but discussions at the time indicated Go would not include species-specific complexes, so things like this weren't included. But so those fell under the Pro umbrella, and at this point, the complex portal was still many years away. Now, the next part of my talk is going to focus on how we automate the creation of Pro terms on a large scale, looking predominantly at UniProt. I've actually never really described this anywhere before, so I thought I'd take the opportunity in this talk. So our first attempt was, as mentioned, UniProtKB. Now, I should note that we did not attempt, and we still don't attempt to ontologize all of UniProtKB. Instead, we focused on what was then 12 key model organisms according to the gene ontology, again, at that time. And furthermore, that includes human, mouse, rat, yeast, E. coli. Furthermore, we concentrated on entries from the reviewed section of UniProtKB, our reasoning being that we wanted to avoid excessive obsoletions, and TrEMBL entries, which is the unreviewed section, are more likely to become obsolete than SwissProt entries. Now, UniProtKB lends itself quite nicely to the task at hand since it contains information on protein name, the organism, what the gene is, etc., along with other leverageable information. And notably, it also has an obsolescence policy similar to those that we at Pro follow. That being said, creating ontological terms for the eligible set is fairly straightforward. And what's less straightforward is dealing with the other types of entries, particularly when it comes to TrEMBL entries and entries that don't conform to the majority organization. To deal with these, it's helpful to understand the potential oddities that one might find, and in turn to understand just why they're odd. So by default, a UniProtKB SwissProt entry intends to describe all protein products of a single gene. When possible, this includes all splice variants, initiation variants, encoded variants, and processed forms. However, due to the occasional inability to describe some isoforms in terms of changes relative to what's considered the canonical isoform, that's the main one that you see if you were to download the FASTA for UniProt, you would see the canonical isoform sequence. Splice variants are sometimes split into other UniProt SwissProt entries. So then we come to the unreviewed cases found in UniProtKB TrEMBL. In a perfectly curated set, all TrEMBL entries that pertain to a gene that's already found in SwissProt would themselves be merged into that SwissProt entry. Again, this is because a SwissProt entry intends to describe all the different isoforms and everything else. As it is, while separate, the TrEMBL entries might describe the same isoform as one found in a SwissProt entry, or it could be a completely different isoform, and it might even represent a variant sequence. So I'll give examples of these and how they're treated in the following slides. So first up is an example of what I call a split entry in UniProt. This is my own terminology. You can tell they're split because they are encoded by the same gene in the same organism, and since both are reviewed, their separateness must be intentional. So the lower panel here indicates why these were kept separate. You'll note the sequences of each isoform are quite diverged at the C-terminus, which is highlighted in red. So how does Pro deal with this? Shown here is a snippet of the hierarchy surrounding the previous example. Since split cases don't have a single main UniProt entry, I should say main UniProt, main UniProt accession that could serve as the parent for all products of the indicated gene, we create one. That's shown. Those three are parent terms that represent all of the gene translation products that could be in those given organisms. Now, split entries can themselves contain either a single isoform or several. If it's single, both the main entry accession and the isoform accession refer to the same thing. By default, since the described entity is always an isoform, we give the term a pro identifier that corresponds to the isoform, so it has this dash number at the end. Finally, though not always necessary, we might create a defined class that holds all the isoforms described within the main entry. So this is what I was talking about as a main accession, is the one without any dash. And this typically would hold all of the isoforms, but in this case, it holds 2 of the isoforms, and the third one is in a different entry right here. So I should say a little something about pro identifiers when it comes to terms based on UniProtKB entries. Pro is a member of the OBO Foundry set of ontologies, and one of the guiding principles is to reuse ontology terms whenever possible. We decided to apply this idea to our use of external identifiers as well. So we wanted to use UniProtKB identifiers fully. That is, if you given the query, it would have the UniProtKB prefix and everything else. However, there was a technical problem with doing this at the time, and I think it still exists, to be honest, that prevented us from using the prefix. So we opted to do the next best thing by using the entry identifier, but prefixing with the PR for the protein ontology. So that's why pro terms have 2 very different looking identifiers. So earlier I mentioned that we, by default, only create pro terms based on SwissProt entries. However, from time to time, people do request terms that are not within this default set, but are nonetheless in the scope of pro. This includes TrEMBL entries. And so next, I turn to the various cases we might encounter with TrEMBL entries. So as mentioned earlier, an unreviewed TrEMBL entry might represent an isoform that's already represented in a SwissProt entry, or it could be an isoform not so represented and is therefore destined to be merged in as a new isoform at some point. Or it could represent a protein from a gene that's not already in SwissProt at all, not even an isoform. Or finally, it could be a sequence variant. With respect to those that are encoded by genes not already covered by SwissProt entries, there are 2 possibilities. The TrEMBL entry is either the only one available, or it is one of several. And if it's one of several, it might either eventually become the canonical isoform, or it could become an alternative splice variant. So at this point, I should describe Pro's dynamic term generator. So if you go to the Pro website, proconsortium.org, toward the top, there's a box that you can use to go directly to the page for the entered Pro identifier. It's a retrieval. But it also has a hidden talent. And that hidden talent is that if you enter a UniProtKB accession, it'll attempt to find the pro term matching that accession, but failing that, it'll attempt to create one on the fly. It'll run through a few UniProt APIs to gather information, and if all is well, it'll output a page providing the requested term, along with a warning banner telling you that it's dynamic and it's not actually in Pro unless you request it, and you have the option to review the OBO stanza. So rather than go through a rather complicated flowchart of how it works, I'll just mention a bit about the queries used and the kind of information obtained. So first, the term generator performs an on-the-fly retrieval of the UniProtKB information based on the input accession. Now, because UniProt has a kind of forwarding address service, a check is done to see if the accession in the returned entry is the same as input. So I mentioned before that TrEMBL entries eventually get merged into SwissProt entries. They become secondary accessions. And so if I were to query with a secondary accession, I would get returned the new accession to which it's been rolled in. Now, queries through UniProt's UniPark database, which is an archive of all sequences based on sequence identity, will indicate which other accessions the requested one might be known by. Finally, using the indicated gene symbol, another query or preferably its model organism database identifier, another query through UniProtKB will indicate if there are multiple possible entries for that gene. Now what happens next is that a few different criteria are used to decide whether or not the entry should be treated as a gene level term or a sequence level term. By default, all TrEMBL-derived sequence level terms are considered isoforms. Unfortunately, that means that cases like the ones shown are not correctly handled because this is a rather obvious sequence variant indicated by the single difference outlined by red. So now, interestingly, while making this presentation, I actually figured out how to solve this problem in terms of determining which is which in a way that doesn't require doing alignments and taking a great time hit. So the issues when dealing with UniProtKB term generation are largely centered around the need to identify the type of sequence that was requested. For the next challenge, which is to create terms from resources that describe proteoforms, the issues differ. For proteoforms, knowledge of the base sequence, that is the correct UniProt entry, is a prerequisite. So too is knowledge of the range of that sequence, at least when the proteoform is one that results from processing the backbone chain. So I refer here to things like chains that result from initiator methionine or signal peptide removal, or by site-specific cleavages that produce mature proteins from longer precursors. So given these prerequisites and what I said before, what could the challenges possibly be? Well, the first potential issue is dealing with disparities in how sequences are numbered, or which isoform is used as a reference for that numbering. For example, Pro, UniProt, and Reactome all use full sequence numbering, that is with the initiator methionine in place, using the canonical isoform, while Alsforum has a preferred non-canonical isoform for one particular protein that they deal with, and Histome numbers its position such that there's, as if there was no initiator methionine. In the latter case, for example, lysine 9 in Histome would be lysine 10 in Pro. The second issue involves the different ways databases indicate modifications. Pro mostly uses the PSI mod ontology for modifications, but uses GNOME ontology for glycans, while UniProt uses its own internal terminology, and Reactome and the top-down repository use PSI mod and KEBI for glycans or special types of linkages. So consider the following example. If isoform 1 of P12345, this is a made-up example, by the way, if isoform 1 is the canonical isoform, and given the ways a phosphorylated residue can be indicated, and given the modified residue is a serine, all of these 6 cases are actually describing the exact same thing. So Pro needs to take care not to inadvertently create multiple terms for the same entity. So that's the big deal with dealing with the proteoforms. So this is my conclusion. So to conclude, the goal of Pro is to harmonize representation of proteins from various databases that have differing ontology usages, but our work requires that these ontologies are mapped, and our use case is yet another that shows how mapping failures can have downstream consequences. So thank you very much.
Great, thanks, Darren. That's a Pro is certainly a ambitious undertaking. I don't know how your wires aren't constantly getting crossed in your mind. I imagine you just dream in all the different possibilities.
I dream in code.
You dream in code, yeah. So I do have one question for you. So looking at the slide where you had sort of the isoforms listed in tree format, and you created different groupings. Yeah, this slide, you create different groupings for, say, isoforms 1 and 2. Are those parent level groupings available? Do you set them up for all of the proteins, or is it set up on an as needed basis? And if it's done on an as needed basis, is there a concern with introducing those groupings later and keeping things harmonized?
Are you referring specifically to this one right here?
To that one, but also to the gene
These two?
translation project. Yep.
Oh, the gene
No,
translation products.
yep. To the
Oh,
higher
okay.
level ones. Yep.
Gotcha. Okay, so we have a way of doing the gene translation product automatically, but I think we tend to do it more on a case-by-case basis. I actually don't remember exactly how we do it anymore, because some of this stuff is coded so long ago that it's lost, it's gone from my memory. But I believe that most, if not all, of the gene translation product guys are done by hand, and but I think that they're found automatically. So in other words, there's a process that we go through each time we run a pro update that shows us where potential problems are, and we address those problems before the update is given. So that's one of the cases.
And it looks like Nico, who's speaking next, has a question. So I'll go ahead and ask it. Do you expect scalability issues when Pro is growing a lot? In particular, do you need OWL reasoners for classification?
So we do actually use reasoners already. In terms of scalability, that's what we're striving for here. There are a bunch of UniProt entries won't scale at all. They need to be done by hand because they don't have genes indicated in them. That's the one of the problems that we have with some UniProt entries. Others are things like fragments, and we don't want to create weird pro terms for, or we don't want to create pro terms for anything weird like that. But we're working towards scaling things, being able to scale up, and that's basically the whole point of this particular talk. The automation part of things is to try and achieve that scalability across all terms that we deal with, including complexes.
Great. Thank you so much again, Darren. This was a great talk.
Thank you.